Assalamu alaikum. Hope you all are fine. So in today's lecture, we're going to discuss the chirality in amino acids. And in our previous lectures, we briefly discussed what are the chiral carbons or the chiral centers present in the amino acids. And we briefly also define what are the chiral carbons or alpha carbons in an amino acid. And just before the start of this lecture, we can have a brief revision of our previous lectures, so that this chirality in amino acids, this concept which is mostly considered to be a confusing topic for majority of the students, so that could be better understandable for you people if we just revise few concepts from our previous lectures. So in our previous lectures, we discussed that there are no doubt there are more than 500 amino acids which exist in nature, and out of these, the 22 amino acids I have discussed which are commonly found in the proteins. But out of these 22, the 20 amino acids are very important because they make up the biological molecules called as proteins, which are found in the human body. And out of these 20 basically amino acids, 19 amino acids are termed as chiral amino acids. Why they are called as chiral amino acids? There is a reason behind it, because they have a chiral center in their structure. And what is a chiral center? All these amino acids have an alpha carbon in the center of their structure, and this alpha carbon at one time is attached to the 4 different groups. Like there is a carboxylic group, there is an amino group, there is hydrogen atom, a functional group, and there is a R chain. And we have also discussed this thing that this R chain is actually different for every 20 amino acid. It could be a particular atom, it could be a molecule, it could be a chain structure, it could be a ring structure, it could be a single atomic form as well, as in case of the glycine, as we're just going to see in a moment. So R chain is different for every amino acid, and R chain is actually there to determine the type of the amino acid, because all these 3 groups are same in the structure of the amino acid. Amino acids are different from each other on the basis of this R group. Okay, so we discussed about the 19 amino acids, which are the chiral one. But what about the 20th amino acid, which is not a chiral one? And that 20th amino acid, that one amino acid is actually called as glycine. And glycine is actually not a chiral amino acid. Why it is not a chiral amino acid? Because it doesn't have a chiral center. And to exhibit a chiral center, there should be an alpha carbon which is attached to the 4 different groups. But in the structure of the glycine, there is a carbon present in the center of the structure. But that carbon is not an alpha carbon or is not a chiral carbon, because this carbon is attached to the 3 different groups. How it is attached to the 3 different groups? It is attached to the carboxylic group, it is attached to the amino group, it is attached to the hydrogen atom, but in place of the R group, there is an atom that is called as a hydrogen atom is present. So its R functional group actually consists of a single hydrogen atom. So that's why now you can see that if I can remove this R from here, because R has been, it's the general terminology which we used. Now there is a H as a R functional group in the structure of the glycine, and now you can see that it is attached to the 3 different groups at one time. That is amino group, carboxylic group, and the third one is the hydrogen, because both the hydrogens are same. So glycine, in case of the glycine, this carbon is achiral. This is not a chiral carbon. So that's why we have just discussed this thing, that glycine is an achiral type of the amino acid, but all the other 19 amino acids are the chiral ones. Now, why we are discussing this achiral and the chiral nature of the amino acids? This is because all the chiral amino acids exhibit a property which is called as optical activity. Now, what is optical activity? Okay, so first of all, let's suppose we have here a normal light. We have normal light here. Okay, and when you pass this normal light through any sort of polarizer, when you're going to pass it through a polarizer, polarizer has the ability to convert this normal light into the plane polarized light. So it will convert this light into the plane polarized light. Now, when you are going to pass this plane polarized light, you can just directly start from here, from the plane polarized light, but I have just explained how the normal light could be converted into the plane polarized light just to make you people understand. But our basic concept starts from here, that when you're going to pass this plane polarized light through any sample that is containing the chiral amino acids. And again, you know that chiral amino acids are those which exhibit a chiral center in them. And the out of 20, 19 amino acids are chiral ones. So here we have a sample that consists of all the basically chiral amino acids. Now, when you're going to pass this plane polarized light through this chiral amino acid sample, it is going to rotate this light. Now your light is going to be rotated either in the left hand side direction or in the right side direction. So if in the sample the majority of the amino acids basically have converted or rotated this light into the left side direction, then that process is called as Levorotation. And the amino acids which have carried out this process of Levorotation, these amino acids are called as L amino acids. L stands for Levorotation. And similarly, if the amino acids present in your sample basically have rotated the light into the right hand side direction, then this process is called as Dextrorotation of the light. Dextrorotation of light, and the amino acids which carry out this process are called as D amino acids. Here you can easily basically predict that it means there are two basic forms of the amino acids that can exist. That is there is one L form of the amino acid that is present, and there is a D form of the amino acid that can exist. And both these forms of the amino acids are called as isomers of each other. Both these forms are actually called as isomers of each other. And in other terms, you can say them both these forms are called as enantiomers of each other. And enantiomers is one of the most important terms you are going to hear again and again in the stereochemistry of the amino acids. So whenever we are going to discuss the stereochemistry of amino acids, you're going to hear this term again and again, that is enantiomers. Enantiomers mean isomers, and there are different types of the isomers of the amino acids. In this lecture, we're going to discuss the two basic types of the isomers of the amino acids, that is L and the D amino acids. In the next lecture, we're going to discuss other two isomeric forms of the amino acids, that is R and S configurations of the amino acids. So just moving forward towards the main lecture, before moving forward, just let me explain one thing here, that is what is stereochemistry. Stereochemistry, from your basic organic chemistry knowledge, you can easily answer this question, that stereochemistry deals with those molecules which have same atomic connectivity. Same atomic connectivity means the number of the atoms in them are same, but arrangement of the atoms is actually different. Arrangement of atoms is different. So stereochemistry basically deals with the molecules which have same atomic connectivity but different arrangement of the atoms. In just a moment, you can just see that what are actually the amino acids which actually fall under this category. Now, in order to have a better understanding of the chirality in amino acids, we are going to study an example which is basically of alanine. And in alanine, again, a central chiral carbon is present, which is attached to the 4 different groups. Here, the carboxylic group will occupy the top position, just like that in the different sugars. We basically place the carboxylic group on the top of the chain. And similarly, as R is actually a variable kind of the functional group, because I have already told you it could be any particular structure. It could be a chain, it could be a ring structure, it could be any atom, any molecule, and it actually defines the type of the amino acid actually. So as it is a variable structure, we place it in the bottom of the chain. Now we are left with the 2 groups. One is basically the hydrogen atom, and the other is basically the amino group. This amino group. Okay. So whenever we discuss basically the chirality in amino acids and L and D forms of the amino acids, we are actually focusing on the position of this amino group within the structure of the amino acids. If this amino group is present on the left side of the chiral center with respect to the hydrogen atom in the structure of amino acids, then again I'm going to repeat this. If the amino group is present on the left side of this chiral center, with respect to what position? With respect to the position of the hydrogen. So it should be present on the left side of the hydrogen or on the left side of the chiral center. Both these things are same. So if it is present on the left side in the chain structure, then it would be L form of the amino acids. And L form of the amino acids are the most commonly found forms in the living systems. Why they are commonly found, I'm just going to explain it in a moment. But let's look on the D form of the amino acids. Now this is another form of the alanine. And here you can see all position of all the groups are same, but the position of the hydrogen and the position of amino group basically has been swapped with each other. Here the amino group is basically now on the right side of the chiral center and hydrogen atom is on the left side of the chiral center. And when this is the scenario, when the you have to just look at the position of this amino group, and when this amino group is on the left side of the chiral center, it is actually the D form of the amino acid. And you can easily now predict from these 2 structures that this is the original structure. It seems like this is the original structure, and this is the mirror image of the original structure. So just to make you better understand, I have just write it here as a mirror, that it is just like the alanine is looking into the mirror, and this is the image of the alanine now, which is its D form actually. So when the position of the amino group is on the left side, it is the L form of the amino acid. Whenever the position of the amino group is on the right side of the chiral center or of the chain structure, it is the D form of the amino acids. Now, to call them as L and D form, one condition is fulfilled. One condition is fulfilled, that is the position of the amino group. You have now understanding of the position of the amino group. But another condition should be fulfilled to call them as L and D forms, true L and D forms. What is that condition? Just look on this hand example. This is your left hand, and this is a mirror image of your left hand. That is, you can say this is your right hand. Okay, so these 2 hands are mirror images of each other. But to call them as L and D forms of each other, they should make what? They should make non superimposable just write me again. Okay, non superimposable mirror images. They should make non-superimposable mirror images of each other. What do you mean by non-superimposable mirror images? Now just just look into this hand structure. Okay, this is left hand, this is right hand, clear. These are the 2 mirror images of each other. And just to make you better understand, we have a diagram of the amino acids over this hand. And now you will wonder that there are the 3 functional groups on the one side of the chiral center and the 1 hydrogen atom on the other side of the chiral center. but this is actual structure of the amino acids, because amino acids exist in the tetrahedral symmetry, or the tetrahedral structure form according to the Fischer projections of the amino acids, and we are going to discuss the Fischer projections just a moment. This is the tetrahedral symmetry of the amino acids, and in the tetrahedral form, the three basically groups, that is the carboxylic group, R group, and the amino group, basically lie on the one side of the chiral center, and the hydrogen atom on the other side of the chiral center. So this is the tetrahedral symmetry of the amino acids, and this is the first structure, and this is the mirror image of this structure of the amino acid. Now, when you are going to superimpose these two images, these two structures, this one left structure and the right structure of one another, and just you can see here like this. Just as you have superimposed your left hand over the right hand, or the right hand over the left hand, in the same way you can superimpose these two structures over each other. And when you are going to superimpose these structures, none of their functional group around the chiral center will superimpose over the other functional group from the same side. Again, I'm going to explain this. When you're going to superimpose this hydrogen will never be superimposing this hydrogen, or it will never overlie over this hydrogen from the other structure. Similarly, the carboxylic group from this structure will never superimpose or will never lie on the carboxylic group from the other structure, right hand structure. Similarly, the R functional group, as in case of the alanine, this is a methyl group, will never lie on the methyl group from the right hand structure. And similarly, the amino group from the left hand structure will never lie on the amino group from the right hand structure. I'm just using left and right hand just to make you better understand. So whenever none of the functional group around the chiral center are superimposing each other, then they are going to make the non-superimposable mirror images of each other. And now they are called as the true L and D forms of each other. First condition to be fulfilled is that the amino group in case of the L form should be on the left side of the chain, and the amino group in case of the D form should be on the right side of the chain. First condition is fulfilled. Second condition is that they should make mirror images, but these mirror images should be non-superimposable. Now they are called as the true isomers of each other, that is L and D forms of each other. So whenever they make non-superimposable mirror images of each other, they are called as enantiomers, and enantiomers mean they are true isomers of each other in the language of what we call as stereochemistry. Okay, so let's just explain one point here, that I have told you that the L form of the amino acids are the most commonly existing amino acids. This is because of the reason that ribosomes basically can easily pick up the L amino acids and can join them together in the form of the strings by means of the peptide bonds to make the long structures and detailed structures which we call as proteins. So it's actually based on the ribosomal activity, that which particular type of the amino acid is actually suitable for them to carry out the process of the translation, that is called as protein synthesis. So as I have already told you in the introductory part, that out of the 20 amino acids, glycine is the one that is actually an A-chiral amino acid. Why it is an A-chiral amino acid? Because it has an A-chiral carbon, or its chiral carbon, its central carbon is not a chiral one. Why it's not a chiral one? Because it is at one time not attached to the four different groups. It is at one time attached to the three different types of the groups. One type is carboxylic group, second type is amino group, and both these groups are basically same. So this is the third type of the groups it is going to attach. So as its chiral carbon is not present in case of the glycine, no chiral carbon is actually there. So that's why it is called as an A-chiral amino acids. And when the chiral center is not present, it means that it doesn't exhibit any optical activity, because The plane polarized light can be rotated in either the left side direction or the right side direction due to the chiral center of the amino acids. And chiral amino acids can only carry out this process. So as there is not a chiral amino acid, so it is not going to exhibit the property of the optical activity. And definitely, if it doesn't have a chiral center, it is not going to make any non-superimposable mirror images. Rather, it will make the superimposable mirror images. How? Let's look into this. So for example, let's just erase this stuff. For example, we have here a mirror image of the glycine. We have a center carbon. It is attached to the 4 different groups. No, it is not attached to the 4 different groups. It is actually attached to the 3 different groups. So when we are going to draw as mirror image, again in the mirror, NH3 positive will be on the right side. Similarly, here will be the hydrogen, there will be the hydrogen, and there will be the carboxylic group. So now, as I have already told you that in case of the Fischer projections, actually amino acids exist in the tetrahedral symmetrical form. That is the hydrogen atom on the one side of the structure, while the other 3 groups lie on the other side. Like there is NH3 positive, there is carboxylic group, and there is a hydrogen. So when you're going to superimpose these 2 structures, just look into this hand example. Just look into this example. So in this particular scenario, let's suppose the R here is hydrogen in case of the glycine. So let me write here, R is actually hydrogen in case of the glycine. So this means that now we have the 2 structures of the glycine. So whenever you're going to superimpose this structure over this structure, then what will happen? This hydrogen is going to superimpose this hydrogen. It will lie just exactly over this hydrogen when you are going to superimpose these 2 structures. So when any one functional group out of the other functional groups around the center chiral carbon are superimposing each other. Again, I'm repeating it. When any functional group around the center carbon is superimposing, then it will not be called as a superimposable form. So when you're going to superimpose these 2 structures over each other, this hydrogen is going to superimpose over this hydrogen. So whenever any functional group around the center carbon is superimposing the other group, the other same group basically in the other structure, then definitely it is not going to make the non-superimposable image. It is going to make a superimposable image, and that is not a property of the L and D forms of the amino acids. So they will make the superimposable mirror images, so the L and D forms basically do not exist. It means that they actually do not exist in the enantiomers form. So out of the 20 amino acids, glycine is basically a exception because it has no chiral center. It doesn't exhibit any optical activity. It makes superimposable mirror images, so its L and D forms are not possible. So in stereochemistry, they are not going to make any enantiomers. Okay, so here we are just going to have a brief explanation of the Fischer projections. I have already told you about the Fischer projections. Fischer projections is just here to explain the relationship of the 4 functional groups. So what is the purpose of understanding the Fischer projections in the stereochemistry? Because Fischer projections helps to highlight the relationship of 4 functional groups of amino acids with respect to its central carbon. So Fischer projections just explain the relationship of these 4 groups carboxylic group, amino group, R group, and the hydrogen atom with respect to this central carbon. So in order to just have a better understanding of the future projections, I'm just going to explain you in a way that, let's suppose this black board is your plane, and on this plane we have this structure and we have this structure. And when we have these two structures, just look at the bonds now. It means that in this structure we have two vertical bonds. These are the two vertical bonds. I'm just going to draw it in different colors. These are the vertical bonds, and these two are basically the horizontal bonds. Okay, so when you are going to see the structure of basically the amino acids in terms of the Fischer projections, you have the two vertical bonds and the two horizontal bonds in the structure of amino acids. And it seems like, now look into this structure. You have an understanding of the vertical and hydrogen bonds now. Now just look into this structure. It seems that these two vertical bonds are coming out of the plane but away from you. And you can have a better understanding of this structure when you will look into the structure of these amino acids from the Lehninger and Lippincott, where they have drawn this tetrahedral symmetry to explain the Fischer projections. So it seems that these two bonds, I have drawn it by myself, but you can have a better understanding of their these two bonds and the symmetry when you will look these structures into the book. So these two vertical bonds are coming out of the plane but away from you, and these two horizontal bonds are coming out of the plane but towards you. And it is when you will look into the structure, it will just show you that these two bonds are coming away from you, or these two bonds are basically coming towards you. So Fischer projections is just here to explain the tetrahedral symmetry of these bonds. So there is a three-dimensional symmetry of the amino acids. They exist in the tetrahedral structure with the two vertical bonds which are coming out of the plane away from you, and the two horizontal bonds which are again coming out of the planes but away from you, sorry, but towards you. So Fischer projection is just to explain the three-dimensional symmetry and the tetrahedral symmetry of the amino acids. Again, you don't have to worry about the amino acids to draw them in the tetrahedral form. You can just draw them in simply in this linear form that I have drawn. In this way, you can just draw the center carbon atom, and then you can draw the four different groups, like you have carboxylic group, you have amino group, you can have any R chain here, and you can have a hydrogen atom. Just you can simply draw them in your notebooks and you're good to go. Fischer projection is just here to explain that you have to remember this amino acids actually do not exist in the planar form. They have this tetrahedral symmetry, this three-dimensional 3D arrangement of the bonds where the vertical and the horizontal bonds are there, and they show certain symmetry with respect to the plane in which they are present. So that's why I have explained the tetrahedral symmetry in this particular diagram, that actually the amino acids bonds exist in this form, and when they superimpose, they should not make any kind of these mirror images. So this was all about the chirality in amino acids, in which we discuss the chirality by considering the one example, that is of the alanine. We discussed the two conditions should be fulfilled. One is the amino group that should be on the either the left side or the right side to be called them as L and D forms respectively. And second is that they should make non-superimposable mirror images. Okay, so these two conditions should be fulfilled. And again, we discuss a particular exception, that is basically the glycine. Glycine is an exception because it doesn't exhibit the chirality, it doesn't exhibit the optical activity, it is not going to make non-superimposable mirror images, rather it makes superimposable mirror images, and definitely it is not going to make any enantiomers. Then we briefly discuss about the Fischer projections. The Fischer projections are just here to explain the exact symmetry of the four bonds of the amino acids around the central carbon, and there are two vertical bonds, two horizontal bonds. Vertical bonds basically are coming out of the plane but basically away from you. Horizontal bonds, it seems like they are coming out of the plane but towards you. So this is how exactly the amino acid structure basically look like, but you can draw them in the simple planar form, and that is all the way acceptable. So again in the let me just show you here again in the tetrahedral form. The amino group in the L form of the amino acids should be on the left side of the chiral center, and similarly amino group here should be on the right side of the chiral center. Okay, so I hope this is clear. If you have any question, you can just write me the question in the comment box and I would be happy to answer them. Thank you so much.